So hi everyone, we're going to go through and we're going to look at the next lesson on heredity. So this whole lesson is how do we actually take our genes that we've found in our DNA and how do we pass them down to our children? Okay, so we're going to first look at some heredity terms. Let's see here. Okay, so remember we looked at karyotypes before in a previous lesson. And so here we have a picture of a karyotype, and there are 46 human chromosomes, and they're arranged into 23 pairs. And you can see here that close up we have a DNA, a piece of DNA, a chromosome, and you can see banding patterns on them. So this is what cytotechnologists use to actually pair up the homologous chromosomes, the banding patterns. And these bands are representative of different genes on different chromosomes. So on each one of these chromosomes in the karyotype over here, you can see that they are going to be carrying different genes. So the gene for eye color might be on chromosome pair number 9, and the gene for hair color might be on chromosome pair number 13. So every single gene is found on one of these chromosomes here. All right. So just close up here, we can see that, I'm just going to choose a different color here. So we can see this gene here is represented by one segment of a chromosome. And in here we might have gene number 1 for hair color and for eye color. Okay, so we might have several different genes on the same chromosome. Okay. So moving forward here, if we can figure out how to do this. All right, so here's a term, allele. So we know that not everyone has the same eye color. So this just means that we have to have multiple mutations or multiple forms for that gene which have arisen because of mutations. So mutations are the source of all genetic variation, which we've already seen in previous lessons of mitosis and meiosis. And you'll also talk about, if you're in the university level course, you also talk about mutations in the evolution unit as well. So depending on what form or of the gene you have, which allele you have for that gene, it can either be dominant or it can be recessive. So dominant is the allele that really takes the cake. It's going to mask the presence of any recessive alleles. So if you have a dominant allele and a recessive allele, then what's going to happen? That's the question. Okay, so what we're going to talk about here are some symbols. So to represent a dominant allele, we're going to use an uppercase letter. Okay, and for a recessive allele, we're going to use a lowercase letter. And this is a way to help us differentiate between the two. Okay. Sorry, bear with me. I'm just learning how to do this too. So going back to fertilization, so we have an egg cell here and a sperm cell here. Remember that eggs and sperm are haploid, which means they each have to have 23 chromosomes, one set of chromosomes. So when we add them together, we get a zygote that is diploid. So we're going to represent that by 2n, or 46 chromosomes total. Okay, so let's keep on with the symbol for eye color here. So let's say that this particular one chromosome, even though we're talking about 23 total, this particular one chromosome is the one that's holding the eye color gene. So let's say that this sperm has the allele for blue eyes, which is recessive, like what we saw up here. Okay, and let's say that this egg cell has a different allele. Let's say it has the allele for brown eyes. Okay, and that's going to be represented by a capital letter, an uppercase letter. So that just means that when this egg and this sperm get together, these two chromosomes are homologous chromosomes because they're both carrying the same types of genes. That means this zygote is going to have two copies for the eye color gene, and it's going to have a dominant and it's going to have a recessive. So that means we have one of each. But what color does that mean? So that means that the dominant allele is going to mask the recessive allele. So this individual, this baby, is going to have brown eyes. Even though it does have a copy of the allele for blue eyes, because the blue trait is recessive, it's going to get masked by the dominant trait. Okay, so now we come to two different terms. We have phenotype and genotype. So up here we were talking about brown eyes and blue eyes. So phenotype, and I remember this because phenotype starts with pH, just like physical starts with pH. So phenotype is really talking about the observable physical outcome of this trait. So if you have the gene for eye color, what actual color do you have? Did you get blue or brown? Or let's say we're talking about the gene for height. Are you going to be tall or short? So this phenotype is really discussing what you can see for that, what characteristic you can see with your eyes that is going to represent the actual combination of alleles. Now the genotype itself, there we go, we're talking about genotype. So this is really talking about what combination. Did you get two dominants from the parents? Did you get two recessives from your parents? Or did you get one of each? Well, these are the only three possible combinations that we're talking about here. So if you got two of the same types of alleles, so that's represented by these two options. You either got two dominants or you got two recessives. This is called the homozygous condition. So this homozygous genotype means you have received two alleles that were the same. So two alleles for eye color that were the same type, the same allele. So either two dominants or two recessives. So homozygous, you can either be homozygous dominant or you can be homozygous recessive. Okay, so there's two options. So you have to differentiate between the two. Oops, sorry. I'm going to do this. Okay, so the other option for your genotype is if you got one of each. And hetero, that prefix hetero means different, and homo means the same. So hetero means you got one of each. And when we're talking about one of each, that means you got one dominant and one recessive allele. So you always, always, always write the dominant allele first. You never ever write your genotype like this with your recessive allele first. This is a no-no. Okay? All right. Let's go back here. So that's a funny little meme there. Heterozygotes. Okay. So here's a nice chart for a scenario. If a child has one of each allele, then what color eyes will the child have? So we've already talked about this. So here's our three possible genotypes. Okay, so we have our homozygous dominant. That's our one type. So if a question is asking you what's the genotype, you can write the actual combination of letters and you can write the description also. So either one is enough to talk about what kind of genotype we're referring to. So we can also have a homozygous recessive. Okay. And the middle combination is our heterozygous option. Now, what phenotype do we have? So remember we said that if an individual has at least one dominant, then dominant allele, its phenotype will be the dominant phenotype. So in this case, in this first case, we have two dominant alleles. So for sure, we know that this individual is going to have the brown dominant eye color. And in the next scenario, the heterozygous individual, because this individual has at least one dominant allele, this individual will also have brown eyes, even though they do carry that blue-eyed allele. And in the last scenario, finally, we get an individual with blue eyes. Because there are no dominant alleles in this homozygous recessive genotype to mask the blue eyes, then finally we have a blue-eyed individual. Okay, so we can see here that even though we have 1, 2, 3 possible genotypes for this trait, we only have 2 possible phenotypes. Okay, so when I post this this PDF PowerPoint file in the announcements, you'll be able to watch the video that's coming next. There we go. So we can see we have a nice video on DNA, chromosomes, genes, and traits. Okay, so you can click the link and go to that and watch it on YouTube. Now, normally in class, I would hand out some worksheets, just some simple practice problems, and we would take it up together. But there are some practice problems like this in your content pages on D2L. Okay, so how do we figure all this out? So back prior to this time period, we didn't know about cells very much. We couldn't even really see cells. Remember that we didn't really discover cells until the mid 1600s. So it wasn't much longer after that where we are finally figuring out what genes are. So take a look at this date, 1822 to 1884. This is when this famous man named Gregor Mendel, he was a monk in Austria, and he spent a lot of his nice quiet days gardening, and he actually grew a lot of pea plants. And what he did is he made a whole bunch of experiments. He did a whole bunch of experiments using pea plants where he bred pea plants together and studied what kind of traits in these pea plants were being passed down from parent generation to offspring generation, and then to the next generation after that. So we call him the pioneer of genetics or the father of genetics, because he was the first one to really discover some of these patterns which we now know to be true. Okay, so what he did is he took 2 pea plants. These are like the plants that produce the green little peas that you eat for dinner. And he cross-fertilized them. So what he did was he took some of the pollen from one plant, and those are produced on the anthers here from one plant. And he took it and he cross-fertilized and made baby plants. Okay, so what he's doing here is when you're cross-pollinating plants, you're mixing genes together. So this is what we are trying to reproduce for sexual reproduction. All right. So what he found is if he crossed a tall pea plant and a short pea plant, what he thought was going to happen was that he was going to get 100% medium-sized plants. That's what he hypothesized, because obviously tall and short mix to make medium. But this is not what he got. He got 100% tall and 0% short. He could have done this experiment 1,000 times, mixing a tall pea plant and a short pea plant, and all of the offspring plants were tall. And he thought, oh my gosh, what is happening here? And he reproduced this experiment again with different traits. Instead of height, he looked at color of seeds. So he crossed yellow seeds with green seeds, and he again found this same pattern. All of the offspring were yellow, and 0 of the offspring were green. So this led him to come to a very important conclusion, which is what we base all of our genetics on at this point. Okay. So this chart, I like doing this on the board in class. I know it's really small here, but I like this because it kind of goes back to what we were talking about with meiosis. So let's say we're looking at these two traits here, or this one trait with two different variations or two alleles. So the tall is dominant, okay, it's represented by the uppercase capital T, and short is recessive, which is represented by the lowercase t. Now, why did we pick t's instead of s's? Well, the rule here is that whatever the tall phenotype is, whatever the dominant phenotype is, for instance, this is tall, we're going to use its letter. So because tall is dominant, we're going to use a T, and short is recessive, so we're not going to use an S. So for each trait, we can only use one letter, a T and a T, an uppercase T and a lowercase t. We're not going to use a T and an S for short, okay, just the same letter. So for tall, what he did was he crossed a plant that was tall, but was a homozygous dominant tall. So that means this plant was a pure breed. It had two dominant alleles. And he crossed it with a plant that was recessive, okay? So to be short, you have to have two recessive alleles, because if you only had one, you would be tall again. So remember that during meiosis, we actually produce 4 gametes. And in each gamete, we have haploid the amount of chromosomes. So what's happening is we can either, this first parent can either donate one large dominant T or another dominant T. Those are the only options that this individual can give. So we just really break these in half. Okay? And the second parent can only give its offspring a lowercase t or a lowercase t. So only recessive alleles. So now I like to think about the next step of this problem a little bit like math, like the FOIL. So I don't know if any of you remember this piece of math problem here where you're multiplying through brackets. Well, so now I have all these gametes. Look at all these gametes. I have 4 gametes that are haploid. So which 2 are going to make the baby? Which 2 are going to be fertilized together to make a baby? Well, we have a number of options here. Okay, so we show the first option. So if the first gamete from parent number 1 mixes with the first gamete with parent number 2, so we show that here with these arrows, then what combination of alleles are we going to get in this diploid zygote? Okay, so this is like FOIL, the first and the inside. So this individual here, this is going to be a diploid zygote. It will have one capital T, or one dominant T, and one recessive T. So what happens if I now mix this first gamete with the second gamete of the second parent? So again, I'm going to get one dominant allele and one recessive allele. Now remember that I'm always writing the dominant allele first. So the next option is we're going to make two fertilizations with the second gamete from the first parent. So we're going to mix this gamete, the second one from parent 1, and the first one from parent 2. And we're going to get, again, a dominant T and a recessive T. And the last one, we're going to mix the second gamete with the first from the first parent and the second gamete from the second parent. And finally, again, we have a dominant T and a recessive T. Sorry, it's getting a little messy, real small. So look what we have. We have 4 possible combinations. Those are the only 4 possible combinations we can make with these 4 gametes. And they're all heterozygous. All of them are heterozygous. And that means that because they're heterozygous, because they have at least one copy of the dominant allele, that they will all have the dominant phenotype. This is why they're all 100% tall. So this explains Mendel's outcome, his results. But he couldn't see genes. He didn't even know what genes were, much less alone what DNA was. So all he could see was tall versus short. So this is why, even though he crossed a tall parent with a short parent, all of those first generation plants in that first offspring generation were tall. Then what he did is he got a little daring. He now took 2 offspring and he cross-fertilized them together. So he took 2 of these offspring tall plants. Okay, so this is represented in the next step. And he crossed them together. And we're going to get a little bit of a different ratio here as a result. So if we took these 2 parents, they're both tall, they're both heterozygous, and they both produce gametes through meiosis. And so what we're going to do is we're going to get some gametes here. So let's just split them up. So we're going to have a dominant and a recessive. And over here, this parent is going to have a dominant and a recessive. Okay, so now we can predict that when we fertilize with these gametes, we're going to get some different results. So let's go ahead and mix these together. So our first option here, we're going to have 2 dominant alleles. You can see dominant. So we have homozygous dominant. Our second option is gamete from parent number 1 and the second gamete from parent number 2. So we have 1 dominant and 1 recessive. And our next option, now we're going to move on to the second gamete from parent number 1. So here we go. And the first gamete from parent 2. And what we see here is 1 of each. And I'm going to write it so that the dominant allele is first. And the last option I have is the second gamete from each parent. Okay, so 2 small lowercase t's, so 2 recessive alleles. So take a look at what we've got here. And just to write them a little bit better underneath, I'll show you. Now look at that. So we have 4 different individuals, but 2 of them are the same. So the 2 in the middle here are both heterozygous, and the 2 on the outside are homozygous. But which ones are tall? Okay, well look at this. This one is tall because it has 2 dominant alleles. It has to be tall. These middle 2 are also tall because they have at least 1 dominant allele, which masks the presence of the recessive allele. This last guy here, because we have 2 recessive alleles, this individual is going to be short. So we can see now here we have 3 out of the 4 that are tall. This is what gives us a 75% ratio here and 25% short. So this really led Mendel to come up with some interesting conclusions. So let's move on to some of his laws that he's come up with. Oh, where are we going here? Nope. We're there. Okay. Bear with me. There. Okay. So Mendel's 3 laws of heredity. So we have a law of segregation, a law of independent assortment, which hopefully you recognize that term, and the law of dominance. So he says here in his first law, when any individual produces gametes, the copies of a gene separate. So this is what we see that's happening during meiosis, during anaphase. So our chromatids are separating, and each gamete is only going to get 1 copy of each chromosome. So this is what we're doing when we're producing haploid gametes. Okay. And then we have those number of variations that we made during crossing over and whatnot. Okay. Now, what the next law is saying is that when we are looking at different traits together, different traits, so one for eye color, one for hair color, one for height, they are inherited independently of each other. So this means that when they are distributed during meiosis, during anaphase, that they are distributed independently and randomly from each other. Okay, so they're not linked. And you can look at each trait individually and follow its pattern. And the last law is talking about dominance versus recessiveness. So he noticed that in his first set of experiments, when he mixed all the talls and the short parents, that there was no medium plants, that they were all tall offsprings, that we have one dominant allele, one version of the gene that is going to mask the recessive allele. Okay. All right, moving on. So now we get to my favorite part in this unit, maybe the favorite part in the course. Genetic word problems. So I know that not all of you love word problems if you're talking about math, but hopefully you'll like these because these word problems follow the same pattern every single time. And if you just follow the pattern, then you'll get it right. Okay, so we use a little chart called the Punnett square. And in your content in D2L, it talks a little bit more about where the Punnett square came from, who came up with it. So this guy here named Reginald C. Punnett, and he came up with this approach for mixing together possible parent gametes and to really predict what the offspring could be. Okay, so we're going to look, I think, at five different types of genetic word problems if you're in the university level. If you're in the college level, you're only going to look at 2, I think, if I recall correctly. Okay, so the monohybrid cross, everybody has to know this, whether you're college or university level. Okay, so we're going to go through this one sample problem together. All right, so it says, consider a cross between a pea plant that is, and what I'm going to do in this question is highlight the important parts. We have one pea plant that is heterozygous for round seeds, and another plant that has wrinkled seeds. Okay, so there's our 2 pieces of information for both parents. The question has to tell you which allele is dominant and which allele is recessive. Otherwise, you cannot do this problem. Okay, and we're asked to find the phenotypes and the genotypes of the offspring, and it wants that as a percentage. So sometimes the question will ask you for a percentage with the percentage sign, and sometimes we'll ask you as a ratio. So we're going to try a couple different kinds. Okay, so you can see here I've set up this question with a number of different steps. So this first step here, that's called the question setup. So you have to write trait and indicate what the trait is we're talking about and in what species. So in this case, we're talking about pea plants, that's the species, and we're talking about round seeds versus wrinkled seeds. So the trait in question is really seed shape or seed texture maybe. So we'll put seed shape in pea plants. If you just put seed shape in what kind of plant, and what are we talking about here? Then you have to identify what your symbols are. Okay, so here we said that the allele for round seeds is dominant. That's why we're using an R, because round is dominant and it starts with R. If wrinkled was dominant, we would use the W's. So R, the capital R, is the round, that's the dominant. And the lowercase r is wrinkled. So remember, we have to use the same letter, not an R and a W. So you would get 1 mark for that, for doing the question setup. The next couple steps are the parental information. So we have to pull from the question what the parent phenotypes are what the genotypes are, and then come up with what the gametes are. So this is a 3-mark step here. So the parent phenotypes, what kind of seed shape do they have? Well, it says that we have round seeds for one parent. So you just put round. You don't actually have to say heterozygous in this step because heterozygous is referring to the genotype, not the phenotype. So the phenotypes are either round or wrinkled. So we have a round parent, sorry, a parent with round seeds, crossed with a parent that has wrinkled seeds. Now we know wrinkled is recessive. So to be wrinkled, we have to be homozygous recessive. But, so we know we have to be this genotype to be wrinkled. But because round is dominant, we could either be homozygous dominant or we could be heterozygous. This is why in the question I had to tell you which version of this dominant phenotype we're talking about. Okay, so I'm just going to go back and erase this here. So this means that the round we're talking about is the heterozygous version. So we have one of each allele. And we're going to cross that with the wrinkled parent who has to be homozygous recessive. So when these two cells carrying these two genes separate during meiosis and divide to produce gametes, what's really going to happen is this individual can either donate a dominant R or a recessive R to its children. And the gametes produced by this wrinkled parent can either be this recessive or another recessive. So even though they're both the same, you have to write both of them, okay? Because it's going to make sense when we put these gametes into our Punnett square. So down here below, we can see a little chart with 4 boxes in it. And these 4 boxes are going to represent the potential combinations for offspring between these two parents. So we're going to label this as F1. So F1 means generation number 1, or the F actually stands for filial generation 1. And we're going to put the parent gametes into this chart. And so it doesn't matter which parent you put at the top and which one at the bottom. So I'm just going to go ahead and put the round parent gametes on the top. So you just assign them to columns here. Then I'm going to take the parents from the gametes from parents number 2 and put them along the side, along the rows. So we can see here how we're setting ourselves up to make combinations of the gametes. So this potential offspring number 1 in the first box can receive a dominant allele from this parent and a recessive allele from this parent. Okay, so we can mix those two gametes together. Now I'm always going to write the dominant one first. Okay, so don't forget to do that. Now in the second box, another possible combination is that I have a recessive allele from parent 1 and another recessive allele from parent 2. Okay, so that individual offspring is going to be homozygous recessive. Then down here in box 3, okay, I'm going to take this this gamete, and I'm going to take this gamete. And then my last option here, I'll do in black. My last option, I'm going to have this recessive gamete and this recessive gamete. Now take a look at what we've got. We've got 2 of the same heterozygous individuals and 2 of the same homozygous recessive individuals. So what does the question actually ask us? Determine the phenotypes and genotypes as a percentage of the offspring. So you're going to put a therefore statement. Okay, so you're going to have therefore the offspring, the phenotypes of the offspring are, and now we're going to just put the percentages. So we're going to put 50% are going to be round, because 2 out of the 4 have at least one dominant allele. And 50% are going to be wrinkled. That's these guys here, because they have the 2 lowercase recessive alleles. And the genotypes of the offspring are okay, and again, we're going to use percentages. So now we just literally copy and paste what the genotypes are. So we're going to have 50% of this heterozygous genotype and 50% of the recessive, the homozygous recessive. Okay, and what you would do, that would be where your question ends. So this would be a multiple mark question. So you'd get 1 mark for this question setup. You'd get 3 marks for the parental information, phenotypes, genotypes, and gametes. Now, don't forget that gamete line, because even though you put your gametes into the Punnett square, unless you have this gamete step, you're not going to get your mark. Okay, and you're going to get another mark for the Punnett square, and potentially 2 more marks for the answer. So this is 1, 2, 3, 4, 5, 6, 7 marks for this type of question. Okay, so I think